So today we're gonna go over cellular respiration. Now, let's start breaking down this title. Cellular refers to cells. Respiration refers to breathing. Now, when you breathe, you take in oxygen and you eliminate carbon dioxide. So it's carbon dioxide, not carbon. Now, when we talk or we take the title as a whole, cellular respiration refers basically at cells taking in oxygen and releasing carbon dioxide. Also, cells to survive require glucose. So basically, cells take in glucose and oxygen and release carbon dioxide, and as a byproduct also release water. Now, this is basically cellular respiration, the short of it. Now, where this process takes place? This process takes place, as we learned before, there is two main organelles that could possibly take place: chloroplast or chloroplast and mitochondria. Now, can it take place in chloroplast cellular respiration? Most likely not, because when we look here, we mentioned that it takes in oxygen and glucose. Now, if it takes in oxygen and glucose, it already has a lot of energy. So chloroplast definitely could not be, because chloroplast is made out of chlorophyll, and then chlorophyll absorbs it, as I mentioned before, absorbs the energy from the sun and turns it into glucose. So this one produces glucose, so basically cannot be part of cellular respiration. Now, the second possible organelle, it's mitochondria. Now, since it's the second one and the only one left, basically cellular respiration takes place in the mitochondria. Now, in the first lecture, or second lecture, or 2 lectures ago, we talked about we talked about mitochondria, and I told you the mitochondria has an outer membrane or inner membrane, and it has these indentations right here, this area right here, which was called cristae. Now, what is the function of the cristae? The function of the cristae, or this indentation, is there because cristae increase surface area. They increase the surface area. Now, it kind of makes sense if you have a line like this or a line that looks like this, which one is longer? Obviously, the second one, right there. The same thing. The inner membrane, it's involved basically in increasing the surface area, and actually that's where the cell respiration, or ATP basically, ATP is produced. It's actually in this cristae where ATP is produced. Now, let's write quickly the general formula for cellular respiration. Carbon 6, hydrogen 12, oxygen 6, plus oxygen turns into carbon dioxide plus water. Now, as a byproduct, we get a lot of ATP. ATP, as we learned last week, it's the energy unit for the cell. Energy unit for the cell. This process takes place in mitochondria. Okay. Now, if you notice here, glucose, oxygen, carbon dioxide, and water. Now, looking at this one, you're gonna notice that glucose, it's broken down to carbon dioxide, and oxygen eventually turns into water. Now, when we look here, the glucose turns into carbon dioxide, we notice that there is one element that's missing for carbon dioxide, which is hydrogen. So basically, glucose, as it's broken down, it releases a lot of these hydrogens. In this case, 12, not 6, 12 hydrogens right there. It releases 12 of these hydrogen. Now, who's gonna pick up these 12 hydrogens? These 12 hydrogens actually are gonna be picked up by the oxygen, which will turn them into water. So that's what happened right here. These hydrogens lost here will be picked up by the oxygen, and then they turn into water. That's how the synopsis of basically cellular respiration looks like when we look in chemical terms. Glucose plus oxygen turns into carbon dioxide and water. Now, as glucose turns into carbon dioxide, it's losing hydrogen and electrons, and the process, it's called oxidation. So basically, glucose gets oxidized inside the cells. Oxygen picks up the hydrogen, picks up the hydrogens there, and the process, it's called reduction. Reduction. So the process, it's called reduction. All right, now let's go over and break down actually this whole process. Now, as you guys gonna look at the slides, I'm gonna keep switching between slides and the drawing, the whiteboard right here. But as you look here, you have mitochondria takes in the oxygen, release carbon dioxide. Cells harvest ATP through the process of oxidation of glucose. Now, glucose, as you notice right here, it's a 6 carbon molecules, turns into carbon dioxide. Remember, there is these hydrogens that are lost. Who's gonna pick them up? These hydrogens come right here, and they're gonna be picked up by the oxygen. And at the end, you will have ATP as a form of energy. Now, when we break down glucose, broken down, or it involves 4 steps, 4 important steps. First step is called glycolysis. Second step is called preparatory reaction, or prep step. Third step, it's called citric acid cycle, or Krebs cycle. And the last one is ETC, or electron transport chain. That's where ATP is produced. And I already told you, where is ATP produced? In the inner mitochondrial membrane. Now, let me break it down and write it down for you guys here. So, step 1, glucose it's going to be broken down to pyruvate. Let me write down here glucose actually too. Glucose. Now, this process, it's called glycolysis. Glucose, lysis, break. Number 2, pyruvate. It's broken down to acetyl-CoA group. This is the prep step. Now let me give you a quick little trick right here so you can understand what happens. Glucose has 6 carbons. As glucose is broken down, ends up as a 3 carbon. Pyruvate, it's a 3 carbon molecule. So basically from one molecule of glucose, you end up with 2 pyruvates. In step 2, this 3 carbon molecule, so there is 2 pyruvates, remember, from here on, from here on down, they take place twice these steps. So pyruvate right there, which is a 3-carbon molecule, it's going to be broken down to a 2-carbon molecule right here. Now, as a byproduct of acetyl-CoA, of a production of acetyl-CoA for second step, we also produce carbon dioxide, which is the 3rd carbon. This is 2 plus 1 carbon from there. They equal 3 carbons. Now, acetyl-CoA moves into the Krebs cycle. So acetyl-CoA moves into the last. Acetyl-CoA moves into the next step, which is Krebs cycle, KC, or citric acid cycle. And what it happens here, this one, it's going to move around through a series of reactions, and this acetyl-CoA is going to be broken down into a few things, which one of them that includes its carbon dioxide. Now, since there is 2 carbon for this acetyl-CoA, this carbon dioxide right there, there's going to be 2 of them, 2 carbon dioxide. Now, steps 4, it's basically electron transport chain. Now, step 4, it's the one that basically uses this byproduct, uses NADH and FADH2 to produce ATP. Now, so far, I haven't mentioned anything NADH, FADH2. What the heck are this one if I never mentioned about them? I'm looking at this whiteboard here. I don't see anywhere NADH and FADH2. What the heck are these 2 things? Now, these 2 are basically transport, the final product. That's how basically these hydrogens that are lost right here, these hydrogens are going to be transported around, attached to 2 coenzymes, NAD+ and FAD. These 2 have the capability to pick up these hydrogens and turn into these transporters, FADH2 and NADH and NADH. So basically, these 2 here are called coenzymes. So these 2 coenzymes here and here, the only function they have is just basically to pick up, as glucose keeps shedding these hydrogens, to pick them up and transport them where? To the electron transport chain. Now, where these NADH are now, because I don't see them in the breakdown on any of these steps, I'm going to go over one by one, and starting with the first step, the second, and the third. So remember, this is the main purpose of slowly breaking down glucose, to release NADH, or to produce, sorry, not to release, to release hydrogen and to produce NADH and FADH2. Now, the first step, glucose broken down to pyruvate. Now remember, the step is called glycolysis. Now, for glucose to be broken down to pyruvate, for glucose to be broken down to pyruvate, it requires one thing. It requires that first we input some energy. Input some energy. Why? Because this glucose is a very stable molecule. Now, for something to be broken down in chemistry, it has to be activated. Input to activate. Now, the only form of energy in the cell, it's ATP. So we're going to input some ATP first to activate it. So first, input ATP to activate glucose. How many ATPs? 2 of them. For each molecule of glucose, 2 ATPs are required for activation. So remember, what I'm going to put above the arrows, it means that it goes inside the cell. What I'm going to put down, it means it comes out. So here, at the beginning, I input 2 ATPs. Now remember, this breakdown of glucose all the way to NADH is at the end requires many reactions. There's about 30 reactions through which glucose is completely broken down to carbon dioxide and water. Now, in the first step right here, glycolysis, you input 2 ATPs. As the reaction proceeds, remember it's 6 carbon. Pyruvate, I told you earlier, turns as a 3 carbon. As it's broken down through this process right here and oxidized, what's going to happen? Eventually, you produce 2 pyruvates. Why? Because if each molecule of pyruvate has 3 carbons, initially glucose has 6 carbon. So if I break, for example, a stick into 2, I need 2 pieces at the end to match the initial size of the stick. So the same thing right here, 6 carbon, and I need 2 3 carbon molecules to end up to 6. Now, during this step of glycolysis, we input 2 ATPs, and by the end, we harvest 4 ATPs. Now, this is the gross product of ATP. What will be the net ATP? Net equal 2 ATP. Why? Because I need to input 2. If I get 4 net ATP for glycolysis, it's 2 ATP. Also, glucose starts being broken down, and now it starts shedding the electrons and the hydrogen. So here at the beginning, we also will have NAD+. We also have NAD+. Now, this NAD+, it picks up those hydrogen that are shed and turns into NADH. Now, there is 2 NADH produced. So there is 2 NADH produced in the first step. Now remember, this is not a form of energy yet, NADH. It needs to turn, to be broken down or oxidized later to actually turn into energy or ATP. So the first step, glycolysis, final product, pyruvate, and along we obtain 2 of these ATPs and 2 of this NADH. Second step, this is called the prep step. Now, make sure you remember, this prep step happens twice. Why twice? Because there is pyruvate. It's the next thing that goes in. Pyruvate, it's the reactant. Now, if you look here, there is 2 pyruvate that you obtained in the previous step. Now, pyruvate, it's going to be broken down to acetyl-CoA, acetyl-CoA. Remember, pyruvate was 3 carbon, acetyl-CoA, as I mentioned earlier, it's 2 carbon. So here what's going to happen, 1 carbon is lost. So how can it be lost? The only way that can be lost, it's in the form of carbon dioxide. Carbon dioxide. Now, let me go quickly back at the PowerPoints, and let's look quickly at the first step right here. Glycolysis takes place in the cytoplasm. So that's one thing that I forgot. It takes place in the cytoplasm, and it's anaerobic. Anaerobic means no oxygen. So a glucose is 6 carbon molecules broken down into 2 pyruvates, because a pyruvate is 3 carbon. Now, the first stage is the energy investment, the one that I mentioned, that you input 2 ATPs. And the last step is the harvesting, where you obtain 4 ATPs and also 2 of those NADH. So if you look at the breakdown right here, this is the summary of the reaction. Now remember, this takes place in the cytoplasm, not in the mitochondria yet. So this step, glycolysis, takes place in the cytoplasm, not the mitochondria yet. You can see it here again. Glucose, which is 6 carbons, broken down further, and eventually you obtain the product, which is 2 pyruvates right here. So you got 6 carbon glucose, 2 times 3 carbons right there. Now, the process through which ATP is produced in the first step, it's called substrate level ATP phosphorylation. And this one, it's an enzymatic process, which means that you use an enzyme. So if you look right here, ATP, it's formed from ADP and a phosphate. Oh, sorry. Yes, ADP and the phosphate right there, they add up. Now, basically, ADP plus phosphate turns into ATP. We learned last week. Now, remember, ATP stored about 7.5 kilocalorie per mole. So this reaction or production of ATP in glycolysis, it is enzymatic reaction. It requires enzyme, and it's called substrate level ATP phosphorylation or ATP phosphorylation. Let's see right here. And here is again, the same one that I showed you earlier, the breakdown. So remember, during the first step, you obtain 2 NADHs, 2 ATP. Now remember, this is the net number of ATP because actually we produce 4 ATP, gross number, but the net number is 2 because we need to input 2 molecules. Now, the next steps, as I started in the previous whiteboard, it takes place in the mitochondria. So everything from here takes place in here, in this little organelle right there. Now, this part right here was called the matrix, and it has all the enzymes required by the reactions. All right, so now let's go to the second step right here. Pyruvate. So remember this one, let me put it right here. Oops, sorry, let me put it right there. Glycolysis takes place in the cytoplasm. Prep step takes place in mitochondria. More exactly, takes place in the matrix. Takes place. So it takes place in the matrix, okay, of the mitochondria. Now here what happens, pyruvate is just broken down to acetyl coenzyme A, which is a 2 carbon, right there. In the process here, there is one more thing that happens. Pyruvate sheds, NAD+. So pyruvate sheds a bunch of these hydrogens. It sheds a bunch of these hydrogens. So what is it gonna do? Turns into NADH. Now, there is 2 NADH for each molecule of pyruvate. For each molecule of pyruvate, there is a 2 NADH produced in the, oh, I'm sorry, it's only 1, not 2, sorry. It's 1 for each molecule of NADH. Now, since there is 2 pyruvates, it would be 2 total NADHs. So for each molecule of pyruvate, there is 1 NADH produced and 1 carbon dioxide. Since there is 2, this is gonna happen twice. Let's go back here and let's look quickly at the breakdown. So if you see right here, prep step occurs twice because there is 2 pyruvates, produces acetyl-CoA, pyruvate transported to mitochondria. Finally, remember, this step takes place in the mitochondria. NAD plus picks up the hydrogen, turns into NADH. Acyl group is attached to the coenzyme A at the end and turn into, that's where this coenzyme A comes from, and it picks up the acyl group. So if you look right here, the breakdown pyruvate turns into acyl carbon dioxide. So one of these hydrogens, one of these carbons turns into carbon dioxide. NAD plus picks up hydrogen, turns into NADHs, and also it has these electrons right there. Remember, as it breaks down, it releases hydrogens, these hydrogen ions plus these electrons that come from it. Now, the next step, it's the citric acid cycle. Now, what the heck happens in the citric acid cycle? Let's go back. Third step, Krebs cycle or citric acid cycle. Now, what the heck happens right here? It's a cycle. Cycle means that it goes and it recycles a certain component. You don't really need to remember that certain component, but I want you to remember what the final products are. First, acetyl-CoA goes in. Now remember, how many acetyl-CoA do we have? 2. Why 2? Because we started with 2 pyruvate. So for each acetyl-CoA, as you break it down, you're gonna get carbon dioxide, a bunch of NADH, and a bunch of FADH2. Now, you guys should be able to tell me how many carbon dioxide do we get? We know that acyl group has 2 carbons. So then how many carbons do you get? Carbon dioxide, sorry, 2 carbon dioxides. Now, as a byproduct here, also we get 3 NADH and 1 FADH2. So remember, there's going to be a bunch of NAD plus and a bunch of FAD that's going to go in here, and that will turn into FADH2 and 3 NADH. Also, for each acetyl-CoA will get also 1 ATP. Now this ATP is also produced enzymatically through the same step as in glycolysis, which was substrate level ATP phosphorylation. Now, since Krebs cycle starts with acetyl-CoA, this whole step will happen twice. So the final product is going to be here what? 4 carbon dioxide, 6 NADH, and 2 FADH2. 2 FADH2. Now let's go back to the PowerPoint and let's see. Let's go back to the PowerPoint and let's see. Oops. Citric acid cycle takes place in matrix, or Krebs cycle takes place in the matrix. Now, acyl group oxidized to carbon dioxide. All this reaction takes place right here. NAD, FAD accepts the hydrogen, turns into NADH, FADH2, right? There's a mistake. The process through which ATP is produced, it's called enzymatic, and it's called substrate level phosphorylation. Now remember, it turns twice. I just showed you because you have 2 acetyl-CoA. So remember, acetyl-CoA, it's twice carbon dioxide, and everything that is here is going to be multiplied by 2, including the ATP, and here also twice, because there is 2 acetyl-CoAs. I keep emphasizing this one so you guys keep track of it. So when you're going to go review it or you study, keep mentioning twice, twice, twice. After pyruvate, it's twice. Is that clear? All right. Now, the last step, which is not very complicated, but a tad bit different. Now, all these NADHs, so let's go here because I have a picture here, a figure here. All these NADHs FADHs, NADHs from all these steps here will end up in the crystal in here, somewhere here in the inner membrane of the mitochondria, where the actual machinery, to produce ATP is. So the machinery, it's embedded here in this inner membrane. Now that machinery, it's called ATP synthase complex, and I'm going to go in a minute over it. But what I want you to take out of these ones, the whole purpose, all these 3 steps so far are for one reason: to produce a bunch of these NADH and FADH and these high electrons right here, these blue dots, which are the electrons. Now remember, if you look around, so far we didn't produce too much ATP. We produced in the first step 2 ATP, and we produced 2 more in the citric acid cycle. That's a total of 4 ATPs. That's nothing so far. I don't have enough to actually survive. I need way more ATPs. So then that's why through evolution, we evolved in such a way that we use this NADH and FADH to complete the number of ATPs that we need. And how the heck this process takes place? Check it out here. See, it shows you right here in the crystal of the mitochondria. There is all these enzymes that we need right there. Now, what happens right here? This hydrogen and this right here. Let me see. I had one here. I thought that I did, but I guess I don't. But right here, next to the inner membrane. Remember, this is the inner membrane where the crystals are. These transporters, NADH and FADH, get to this site right here, to these sites right here, next to these proteins. This blue stuff, these blue squares, these blue circles here are actually proteins. So this NADH and FADH come here, and what do you notice? They dump the hydrogens. They dump these hydrogens here at this position right here. So now what's going to happen? Basically what's going to happen, NADH turns back into NAD plus because it dumps this hydrogen. This NAD plus, what is it going to do? It goes back to pick up more hydrogens. Same thing for FAD. They go back to pick up more hydrogen. Now here what's going to happen, these hydrogens have to be pumped out. Where? In the intermembrane space. Have to be pumped out. Now, where is that intermembrane space? It's right in here, guys. You see right here? That's where the intermembrane space, that's between the outer and the inner membrane. So these hydrogens are pumped. Now, where is the energy coming from? As you may notice right here, from this electron. That's why these electrons are needed. These electrons here have high energy, a lot of energy. So then they are able to actually pass on or use that energy to basically pass the hydrogen across from matrix to the intermembrane. So why do we need energy? Because it goes against the gradient. If you would be like this, let's assume this is the inner membrane, inner. This is the intermembrane. If you look right here, here you have very few hydrogens, but here you have a ton of hydrogens, a ton of these hydrogen ions, a ton of them. Now remember, this is hydrogen ions, not hydrogen. Hydrogen is a gas and it's toxic. We don't want that. We have hydrogen ions. Now, hydrogen ions are moved from low to high concentration. To move from low to high, it requires energy. That's why the energy of the electron or the electrons release their energy and help to push these hydrogens across, so they go from low concentration to high concentration. Now, as hydrogens end up in the inner membrane, it creates a really high gradient right there, really, really high concentration. Now, if you keep building them up in here in the middle, right here in the intermembrane, it's a problem because they're going to keep building up and eventually they become toxic and cellular respiration will not take place, or electron transport chain will not take place. Now, this step is called electron transport chain because you notice here, these electrons are transported between these molecules right here. And as they are transported, they go from high energy right here to low energy. Once they get right here, they have no more energy. But as they get passed along this machinery here, they pump the hydrogen. They pump the hydrogens in the intermembrane space. Now, as I mentioned earlier, you have a really high concentration here. Now they have to come back or escape somehow. Now they cannot escape outside. They cannot go outside. They can't, will not go outside. They have to come back through this machinery right here. This machinery is called ATP synthase complex. And it's made up of a few proteins. And I'm going to show you at the end a video how it works. And I'll recommend you guys to watch to understand. But there is thousands of these machineries in one mitochondria. In one mitochondria, there is thousands of these machineries which have the ability basically to allow hydrogen to come back. And while hydrogen comes back, it produces ATP. Now, this machinery looks like a shaft. A shaft is like in an engine, like a rotary engine in the car engine. As the shaft gets rotated, as the shaft keeps rotating, or it spins, it produces energy. It produces energy. While it produces energy, this energy cannot be turned directly into ATP. So what happens, as it spins, there is a little protein right here which will pick up this ADP and a phosphate, puts both of them in here, the ADP and the phosphate, and then it will turn into ATP. It will turn into ATP. So as this machinery constantly spins, and remember, there is thousands of these ATP synthase complexes in the actual mitochondria. As this machinery keeps spinning, it keeps producing energy. There's a bunch of ADP in there and a bunch of phosphate, which will turn into ATP. And I show you in a second a video with it. Now, remember, so far we just mentioned these electrons are here. Once they get here, they have no energy. Now also, these hydrogens come back. These hydrogens come back. That's a problem. Because now they come back here and I have to start getting rid of them. Now, the only way you can get rid of them, it's actually on this part right here, the last protein of this electron transport chain. That's where basically hydrogen ions binds with oxygen. So hydrogen and oxygen bind to turn into water. And then water just moves out of the mitochondria and it can be eliminated through breathing or through skin, or it could be used inside the cell. Now you can see here the enzymes, they talk about them. I don't care. You don't need to know the cytochrome. You don't need to know the cytochrome. You don't need to know the protein complex. You don't need to know anything. All what I want you to remember for the purpose of this class is that this NADH, actually, let me put it in. Let me do it on a drawing so you guys can do it yourself at home and try to do it yourself at home. So this is the inner membrane. Remember, this is the inner membrane. This is the outer membrane. This is the intermembrane. Right here, remember. Now right here, remember that you have this lipid bilayer, okay? Now, what happens right here, basically, you have these proteins. You have these proteins here. Now, NADH comes here. FADH, there is another place to moor or to come to. Once they get here, this NADH dumps the hydrogens. This FADH dumps the hydrogen ions. Then the hydrogen ions are pumped in here. With the help of what? With the help of these electrons. Remember, these electrons that they're gonna start moving this way. This is the electron that's going to start moving this way. Both of them have electrons that have energy. This hydrogen, it's also moved this way. So we get a really high gradient of hydrogens in here, hydrogen ions. Now these hydrogen ions come back through this machinery that I talked to you earlier. This machinery that has wheels right here, I'll show you in a second. So hydrogens come back, and as they come back in here, this machinery spins. This is the ATP synthase complex, ATP synthase complex. So what it does, ADP here plus phosphate turns into ATP. Now this hydrogen, it's brought to this side right here, where hydrogen binds to oxygen and turns into water. Now on Friday, when we're gonna go over the lab, or you guys gonna go over the lab for Friday, you're gonna notice actually examples how chemicals or how poisons actually can affect us or can kill us. One of the poisons that can kill us is cyanide, that's used for rats, to kill rats. Now, cyanide actually comes and blocks this reaction from taking place. So if I block this reaction, I keep building up these hydrogen ions right there, and I'm gonna die from it. That's how basically we're gonna die, because we get, or the rats get, high concentration of hydrogen, which basically cannot escape. So remember, last time, NADH and FADH2, we did their electrons, the high energy electrons, come here. They're going to dock, each one on their side, release the hydrogens, release the electrons. Hydrogens are pumped across the membrane from low to high concentration. The energy to move them across, because you go from low to high, comes from the electrons, these ones, which will move along the way and keep pumping these hydrogens across. Hydrogens then come back through ATP synthase complex, and then that machinery spins, and the energy is used to bind ATP and phosphate into ATP. The hydrogens, as they come back, they go directly to this side. Now, some of you might ask me, oh, well, why do we have to pump them between the intermembrane and then bring them back in the matrix again? Because this part right here, this is the matrix. Why would I pump them from matrix out and then bring them back? Because that's the only way we can produce ATP. These electrons, they have high energy. We'll never bind here to oxygen because they're too much energy. These hydrogens right here could bind, but there is a little wall right here that blocks them from actually reaching the site where the reaction will take place. So then the only way to get to the site is like going around. So it has to go through the outer and come back through the inner part right here. Now, at the end of electron transport chain, we produce 34 ATP from all the NADH and FADH that's available there, or it came with it. So far, let's see. In first step, we produce 2 ATP and 2 NADH. Now, in theory, and please just remember this one, I'm giving you whole numbers rather than give you different numbers, like half a number or quarters of a number or anything like that. For each NADH, we get 3 ATPs in the electron transport chain. For each FADH2, we're gonna get 2 ATP. So now let's do the math. In glycolysis, we got 2 ATPs plus 2 NADH. This one turned into 6 ATP. 2 times 3, each NADH is 3 ATPs. Second step, we had 2 NADH, so we had 6 more ATPs. Third step, Krebs cycle, we had 2 ATPs produced, plus we had 6 NADHs. If you go back and you look at it, 6 NADH, which equals to 18 ATP. Also, we had 2 FADH2, so 2 times 2 ATPs equal 4 ATPs. So now when we do the math, let's see how many do we have. 4 plus 18, 22, plus 2, 24, 24 plus 6, 30, 30 plus 6, 36, plus 2, 38. So we have a total of 38 ATP. Now in the books, you guys are gonna find 36, 32, a bunch of other numbers. Don't go by those books. I'm giving you this number as a whole number to simplify the life. Are we clear, everybody? So please follow my advice or whatever I tell you. Now let's go here, PowerPoint, and see it. Oops. Let's go right here, share. So now you can see here the electron transport chain again, and you can see the ATP synthase complex. You can see the machineries right here, citric acid. Look at other pictures online if you want, videos, whichever one helps you. Here is the total, 3 ATPs for NADH, 2 ATPs for FADH. Takes place right in here. I have another good picture right here. This is the machinery basically of ATP synthase complex. As hydrogens move in, this rotary machine, this shaft right here spin, and that's where ADP and phosphate come in to produce ATPs, and hydrogens escape through there. This is another picture of how complicated this machinery is. Look at these proteins right here, how complicated you are. You see, this is the place where the NADH docks right there. I don't see it right there. This should be the place where the actual FADH docks with it. I don't see that it docks right there. Oh, maybe I know, I don't see it right here, but that's what it happens right here. Okay, that's where FADH2, that's where the FADH2 is going to be used. Okay, again, you can see the flow of electrons, ATP production, 38 total right here. 38 total. You can do your own math. Here I put there the actual number. It's only about 29 to 30. Why? Because there is a bunch of other things that are needed there. So if things are needed, you need to use energy. I'll show you at the end the video. I recommend you the video to watch. Fermentation is the next step. Now, so far, I mentioned that glucose turns into pyruvate, and then it moves forward into Krebs step, Krebs cycle, and electron transport chain. For these steps to move forward, it requires oxygen. It requires oxygen. If there is no oxygen, it's gonna move into fermentation. It moves into fermentation, which is anaerobic, or it means there is no oxygen. So to move towards Krebs step, Krebs cycle, you require oxygen. And remember, the oxygen is required because on this step right here, to pick up the hydrogen to turn into water, to eliminate those hydrogen ions, which are toxic. Now, if you don't have oxygen, like for example, when you go running, you slowly start running out of oxygen. Why? Your cells are starved from oxygen. Why? It's not you. You don't really run out of oxygen, but you run out of breath. Why? Because your cells requires a lot of oxygen, but as you run, they work harder and harder. They produce more and more of those hydrogen ions, but you don't get enough oxygen because you cannot take it in. Now, what's going to happen? You basically, your whole body moves into this process called fermentation. And all of you felt this one. If you went running and you're not trained, you start getting this really burning, like really burning in your muscle. Why? Because you start producing lactic acid. Now, during this process of fermentation, basically your cells survive only on the 2 ATPs, those 2 net ATPs produced in glycolysis. Now what happens, or why do we go in this step right here? Because remember, we produce 2 NADHs in the first step. Now you can get rid of these hydrogens. You can unload them. Where? Somewhere here at the end in electron transport chain. You can unload them because you can move forward. So then the body shifts this way, and this NADH unloads the hydrogen, which turns into it's used to produce lactic acid, and then the NAD plus, it's returned back into the first step. Why? Because NAD plus turns into more NADH. As glucose gets broken down, more of those hydrogens are released. Somebody has to pick them up, and who picks them up in the cell? NAD plus, nicotinamide diamine. That's the one that's going to pick them up, the NAD plus. So the whole purpose of this fermentation is to recycle the NAD plus. Now, if you're done running, you stop. You still have your muscle burning. Now what's going to happen? It's basically the reverse. Lactic acid, it's taken back to the liver from the muscles, and it's turned back into pyruvate or into glucose. Now this process, it's very slow. It takes a few days. You know, when you get muscular fever from running or that burning, it takes a few days to go away. Why? Because it's very slow process. All this lactic acid has to be taken back to the liver, where it has to be turned back into pyruvate or glucose. Now, this is the fermentation that takes place in our muscles. Now, how about the good old fermentation? This is the one, again, the one that I just discussed. The process is, the purpose is to recycle the NAD+. How about a good old fermentation to produce alcohol? It's exactly the same thing as this one right here, glucose broken down to pyruvic acid. And then what happens for alcoholic fermentation? Rather than producing lactic acid, it produces alcohol. Same thing. The purpose is to recycle this NAD plus to go back into glycolysis. Now, what's really cool is that these bugs called yeast cells actually do not need more than 2 ATPs. 2 ATPs are way more than enough for these bugs to produce alcohol. They don't need 38 for each glucose molecule. So yeast, it's okay, and will survive with 2 ATPs only. Now, alternative metabolic pathways. So far, we talked only about the main one. And remember, glucose is the main energy for the cell. Cellular respiration will not happen if there is no glucose or you are starving from glucose. So glucose is very, very important in our diet. Now, if you look at the breakdown of cellular respiration, you notice that carbohydrates or sugars enter right here. Now, fats, if you notice here, can be broken down into fatty acids and glycerol, and we learned this one in the second lab. Fats, these are the components that make up the fats, and glycerol can enter in glycolysis and at acyl-CoA. So fatty acids can be broken down to acyl group through beta oxidation, and they can enter at 2 steps in cellular respiration. But remember, we require glucose for that. Proteins, they're made up of amino acids. Amino acids can be broken down into the different components or turn into different components that can be used to produce energy. So that's why your body needs all 3 sources of energy: carbohydrates, fats, and proteins. They are used a little bit different and stored around the body, and also the main product or the main function of all of them, or the most important function, is to help us survive by producing ATP. So remember, carbohydrates enter only at the beginning, sugars. Fats can enter at 2 steps, and proteins at 3 steps along the cellular respiration. Now, quickly, just a case here. What is the difference between a marathoner and a sprinter? Now, the process or the difference between somebody that runs marathon, which is skinny, very skinny people, and then a sprinter, they're kind of like solid built. Think about Usain Bolt, and then think about one of those African runners that run the marathon. Africans are very skinny. but they can run for 30, 40 kilometers, right? 45 kilometers, the marathon, and they have no problems. Now, Usain Bolt is a great sprinter, but if you tell him to run the marathon, the guy will not probably survive even to 10K or 10,000 kilometers, 10,000 meters or 10 kilometers will not survive or not even compete with those Africans. Why? Because it's different is in their muscles, basically the type of fiber they have and how they use that to produce ATP. So basically the difference between the marathoner and sprinter is the process that each uses to make ATP. Now, if you are marathoner, you have slow fibers. This one, what they do, they aerobically, or they evolve, or they, as you keep training, evolved in such way they aerobically use oxygen to produce ATP. Then the fast fiber use it anaerobically. Basically, what it means, they don't use oxygen. So the slow fibers, they evolved that they aerobically use oxygen. Fast fiber, they use anaerobically oxygen. Now, the percentage of slow and fast fibers that we have, that's what determines what type of athletes we are. Usain Bolt, remember, it's very thick, a lot of these fibers that he has right here. So basically, anaerobically, he produces about 2 ATPs. And with those 2 ATPs, he can run only short distances. Now, if you have slow fiber, you're a little skinnier, and all these fibers in here produce 38 ATPs because this one is aerobic and this one is anaerobic. So remember, the difference is when you go out there and try to run, it's how you train. If you train yourself since you're young for short distance, for sprint, you're gonna be more muscular because you build more of the fast fibers. If you wanna run the marathon, you build more of the slow fibers. And you could see them right here, how they look like. And this is the whole lecture for today.